Hi everybody and welcome to this, which is lesson 1 in section 1.6, all about mutations. And in this lesson we're going to take a look at gene mutations. So we're going to describe what a mutation is at high level, we're going to describe the different types of gene mutation and the effect they have on the protein produced. By the end of the lesson I want you to be able to describe the 3 different types of gene mutation and identify the impact that they have on the polypeptide and then ultimately the protein produced. I also want you to be able to describe what a splice site mutation is and the impact of splice site mutations on an organism. So we took a look at mutations at Nat5 level, and at Nat5 level we tend to say that a mutation is a change in the DNA, and mutations are what give rise to the variation that we see within species. And that's true still at higher. Okay, within the human population there are lots of different types of variation in the genomes, and that is the result of mutations. So remember, at national 5 level we talk about the fact that things like eye colour differs within the population. So some people have brown eyes, some people have blue eyes. Those people who have brown eyes, the gene is functional and it's producing that melanin pigment which is making their eyes have that brown colour. Those people who have blue eyes, that gene is mutated, it's not producing that protein pigment, and therefore we have a different characteristic. Okay, and you can think about that in terms of lots of characteristics across a population. Now this is our definition at higher level. So mutations are changes in the genome, and we learnt that word last lesson. Remember, genome is all the hereditary information encoded in an organism, and that results in no protein or an altered protein being expressed. So remember, our genes are the sections of DNA that will code for proteins. If there is a change in that coding section, then obviously we're going to get a change in the product released at the end, which is our protein. And 3 words that we use to describe mutations is that they are spontaneous, that they are random, and they are rare. So they can happen at any time to any organism within the population, but they occur very, very rarely. And on our first exercise, we're going to take a look at just how rare mutations are. So mutation rate can actually be calculated. Now, generally, when we're calculating mutation rate, we look at the number of mutations that occur on a particular gene site per million gametes. And again, just to recap, a gamete is a sex cell. So we have a data table here. And in the first column, you can see we've got 4 genes that have been mutations. So remember, we represent the genes by the letters. The capital letter means that it's the dominant form, the lowercase letter identifies that it's the recessive or mutated form. So we've got 4 mutations here. So big R being mutated to little r, big I being mutated to little I, big SU being mutated to little su, and then big SH being mutated to little sh. In the second column, you can see how many gametes were tested to see if they had that mutation present. In the third column, you can see of those gametes tested, how many of those gametes had that particular mutation. Now, the first thing I want you to do is I would like you to copy the table down and I would like you to complete it to show the frequency of the mutation. So in order to do that, if you think about that first mutation, big R to little r, we need to take the number of mutations and then divide that by the number of gametes tested to find out on average how many mutations have we got per gamete. Then to convert that into mutations per million gamete, you need to multiply that number by 1,000,000. Once you've done that and you've completed your table, I would then like to have a think about the next 2 questions. So why is it necessary to convert to per million? And then secondly, what does this data tell you about mutation frequency? So if you'd just like to pause the video and have a go at that task, once you've unpaused it, I will go through this with you. So hopefully you got the following answers. Okay, so in our first gene, the average number of mutations per million gamete was 492. In our second gene, it was 106. In our third, it was 2. And in our fourth, it was 1. Now, why is it necessary to convert to per million? Well, obviously, if you're looking at that second column, the number of gametes tested each time is completely different between the 4 genes. In order to make that data more comparable, we need to convert it to average number of mutations per million gametes. Okay, so the number that we're testing is being kept standardized. We can't compare the data in the third column because the number of gametes tested has varied throughout. And then lastly, what does this tell us about mutation frequency? Well, it's just reaffirming that point that mutations happen very, very rarely. So what we're going to take a look at next is we're going to describe some gene mutations, and there are 3 different types of gene mutation. What a gene mutation is basically what is happening is there's going to be a change in one of the nucleotides within the DNA nucleotide sequence. Okay, so you can see in this little picture, in that original DNA sequence, that nucleotide that had the thymine base has been altered, and a different nucleotide has taken its place, one with a cytosine base. Please remember that when you're describing gene mutations, please talk about the fact that it's a nucleotide that is altered, not just the base. Remember, the base is only part of the whole nucleotide molecule. In a gene mutation, it's the nucleotide that is being altered. And there are 3 different types of gene mutation, and I like to remember them using SID. So our 3 different types of gene mutation are substitution, insertion, and deletion. And you'll see I've put in a couple of other keywords linked to these 3 gene mutations. So our substitution mutations can be further categorized into what's called a missense and also a nonsense substitution. For our insertion and deletion mutations, they are known as frameshift mutations. And what we'll do is we'll work through each of these gene mutations at a time, explaining what those keywords mean. Whilst we're working through, I want you to be pausing the video and having a go at the activities. You will need your mRNA codon table. So if you do not have this codon table in front of you, take a screenshot on your phone of this table and use it as we are working on the task. Remember that how to use this is that when we've got our mRNA codon worked out, we work from the centre outwards, okay, to read that codon. So if the codon is ACG, the amino acid that would be coded for would be threonine. Okay, so you always work from the inside outwards on this particular table. So take a screenshot of that and we'll use it throughout this task looking at these different gene mutations. Now the first gene mutation that we're going to take a look at is substitution, and it does exactly what it says on the tin. So in a gene substitution mutation, one nucleotide, remember a nucleotide, not just a base, is replaced with another nucleotide, but that other nucleotide contains a different base. And now we're going to take a look at some examples of substitution mutations. So what I have here is I have an unmutated DNA sequence. So TAC, ACG, CAA, AAT, ACAACT. Now what I want you to do is, first of all, I would like you to transcribe this DNA. Now remember, during transcription what happens is that DNA sequence is opened up by our RNA polymerase, and our RNA polymerase through complementary base pairing will join together our RNA nucleotides to form our well, initially our primary transcript and then our messenger RNA, but we're going to, just skip a step and make sure that we're producing the mRNA. So the first thing I want you to do is I want you to work out the sequence of RNA bases on the mRNA. Then secondly, using your mRNA codon table, I want you to analyse the mRNA that you have, and I want you to translate it into the sequence of amino acids. Now remember, you don't have to write down the whole amino acid name, you can just use the first 3 letters. So for example, threonine would be THR. Okay, so again, pause the little video, and all I want you to do is just transcribe and translate that sequence of DNA. So hopefully you worked out the following. So this is our mRNA sequence. So TAC, remember that where there is a T, we would have complementary adenine. Where there is an adenine on the RNA, remember thymine does not exist, so we would have the uracil base coming in. Okay, where there is a cytosine, we would have the complementary guanine, and so on and so forth. Okay, so your mRNA code should be AUG, UGC, GUU, UUA, UGU, UGA. Okay, and just for the ease of the next step, I've split the mRNA codons, I've split the mRNA sequence into the codons. So you can see we've got 1, 2, 3, 4, 5, 6 codons here. And then for our amino acid sequence, as always, our amino acid sequence is starting with our start codon, AUG, so therefore our start amino acid is methionine. UGC would be coding for cysteine, GUU would be coding for valine, UUA would be coding for leucine, UGU would be coding for cysteine, and then UGA would be our stop codon. Remember to please write in the stop, don't just leave it blank. So that is our normal unmutated DNA sequence. Now the next thing I want you to do is, I've given you here an example of a substitution. So here in this particular DNA sequence, that sixth nucleotide has been substituted for a nucleotide with, rather than cytosine as the, guanine as the base, it has been replaced with a nucleotide that has thymine as a base. So again, what I want you to do is I want you to pause the video and I want you to work out what is the impact of this on the polypeptide produced. So transcribe it into the messenger RNA, then using the codons, work out what the polypeptide chain would be. So hopefully you found the following. Our mRNA codons are almost exactly the same. The only one that has changed is that second one. It's gone from UGC to UGA. However, our polypeptide chain has changed quite significantly. We have methionine, which is our amino acid that starts the polypeptide chain, but then suddenly, rather than cysteine being coded for, we have a stop codon very prematurely being coded for. So what does this mean? Now, this particular substitution is an example of a nonsense substitution. What has actually happened is a premature stop codon has been produced. Now, have a little think. What is the effect of that on the polypeptide? And then also, what do you think would be the effect of that on the protein? Again, pause the video, have a little think, form your answer, and then listen to what the feedback is. So hopefully you can see from the previous slide that the effect on the polypeptide, it would be greatly reduced. Okay, if a stop codon comes in prematurely, no other amino acid can bind on. Okay, remember that stop codon brings in no amino acid at all, so therefore no further amino acids could join because there's nothing to form a peptide bond with. The effect on the protein is that our protein would be shorter, and it would also be non-functional. There wouldn't be all the amino acids needed to help it to fold correctly, to form its function, so therefore we'd have a shorter polypeptide, we'd have a shorter protein, we'd have a non-functional protein. In the next example, we're still going to look at a substitution mutation, but again we've got the same DNA sequence and the same polypeptide sequence, so this is the one that is not mutated. Now, I want you again to have a little think using your mRNA codon table, what would happen with the following substitution mutation? So here in that second sort of DNA triplet, that guanine base has been replaced with a nucleotide with a cytosine base. So again, pause the video, transcribe this DNA into your mRNA, and then using the mRNA codons and your mRNA codon table, have a look at the effect of this particular substitution on the polypeptide chain produced. So if you want to pause the video and have a go at that, please do so now. Hopefully what you should see is if you transcribe that DNA into the messenger RNA, we get the following mRNA transcript, and then this is what happens to our polypeptide chain. So what has happened here is we haven't got a premature stop codon being coded for. Instead, what's happened is where that substitution mutation has happened, the amino acid that was previously coded for was cysteine, and what's happened here is a different amino acid has been coded for, which is tryptophan. Now, this particular substitution mutation is known as a missense substitution. And here, one amino acid has been changed for another, a completely different amino acid. Now here, obviously, the length of the polypeptide chain hasn't been changed at all. All we've done is we've changed one amino acid out of sequence. And I want you to have a think about what effect that would have on the protein. And what I want you to remember is that in a polypeptide chain, the sequence of amino acids is really quite crucial, because that sequence of amino acids in that polypeptide chain will influence the folding that happens. So remember that initially we have our polypeptide chain and hydrogen bonds will form that will cause it to fold initially. Then other types of bonding will cause it to fold even further. Now, one amino acid changing out of sequence could mean that actually that particular polypeptide chain doesn't fold correctly into its 3D shape. If that's an enzyme, for example, and it doesn't have the correctly folded active site, that means that that particular enzyme is not going to be functional. It's not going to be able to synthesize or degrade any sort of reaction. So it will really impact on that particular protein's function. Sometimes, however, the amino acid that's replaced for doesn't have an impact on the bonding, okay, and actually has no effect on the protein at all. So with this particular substitution mutation, it depends, okay. It could potentially cause the protein, cause the polypeptide chain not to fold correctly and therefore the protein not to function correctly, or it might have a very, very minimal effect. So that is our missense substitution. Now the next gene mutation that we're going to take a look at is a deletion mutation. Within a deletion mutation, what will happen is we will have the loss of a nucleotide from a sequence. So again, we're going to take a look at what effect this has on our polypeptide chain that's produced. Still working with the same DNA sequence, still working with this normal sequence where the polypeptide chain produced is methionine, cysteine, valine, leucine, cystosine, and stop. Now what's happened is there has been a deletion mutation and the following DNA sequence has occurred. Now all I want you to do is, same as usual, without thinking about where the deletion mutation has occurred, I want you to transcribe that DNA sequence into the messenger RNA sequence and then translate that mRNA sequence into the sequence of amino acids in the polypeptide. So again, pause this video and then using your mRNA codon table, have a go at that task. Now what you should find is the following. So if we transcribe it to the messenger RNA, now you'll notice that because there's been a deletion mutation, our last mRNA codon is only 2 letters long. But you also might notice the following. When you translate that mRNA sequence into your polypeptide sequence, where the deletion mutation happened, which if you were looking very carefully, happened in that second codon along within the DNA triplet, the guanine, or the nucleotide with the guanine base attached, was deleted out. Where that deletion mutation happened, every single codon and therefore every single amino acid after that particular mutation has been affected. Okay, so the only amino acid that is similar from our normal DNA and compared to our mutated DNA is that methionine, that very, very first start amino acid. Every single amino acid coded for afterwards is completely different. Now the last one we're going to take a look at is called an insertion mutation. And again, very, very straightforward. An insertion mutation is just where a nucleotide is added into the DNA sequence. Now I would like you to try the following. So there we have again our unmutated DNA sequence with our normal polypeptide chain produced and our sequence of amino acids. Here we've got that DNA sequence with an insertion mutation added in. Okay, so same as before, I want you to transcribe the messenger RNA code from the DNA, and then I want you to translate from that messenger RNA code into the sequence of amino acids that would appear in the polypeptide. So again, pause the video and have a go at that little task. Now hopefully you should see the following. So again, we've got a change in the messenger RNA codons, and again that last one, because we've had an insertion mutation, our last codon is only 1 nucleotide long. And again, where that insertion mutation happened, which was, if you're looking at your DNA code above, okay, where that ACG triplet is, that second one along, after that we had a nucleotide with adenine added in. Okay, so you can see that just about there. So there's where that nucleotide has been inserted. Now, as you can see from your messenger RNA, every single codon after that particular mutation, and therefore every single amino acid that occurs after that mutation has been affected, and they're all completely different, which is very similar to what we saw with our deletion mutation. So where the deletion and the insertion occur, every single codon and every single amino acid has been affected. And this has a name. Now, this particular effect that we can see, where every single amino acid and every single codon after the mutation is affected and changed, This is known as a frame shift, and this will have a major effect on the protein produced. So the sequence of amino acids after that mutation will all be completely different. So that's going to have a significant impact on the bonding that occurs, that will influence the folding that occurs within that protein structure, and ultimately how that protein functions. Now, the last thing I want to talk about is splice site mutations. And as it is quite aptly named, splice site mutations occur during that splicing process. Now remember, splicing is something that takes part after transcription has occurred. So remember, our DNA will unwind and unzip. So the RNA polymerase will unwind and unzip the DNA, and it will start building that primary transcript. That primary transcript, remember, will then undergo RNA splicing. So the non-coding sections of the DNA, the introns, will be removed, and the exons will be spliced together to produce messenger RNA transcripts. Now, what can happen is during that splicing is that some introns could be retained, they're not actually removed from the transcript, or some exons are not included that should be. Now remember, I know that with alternative RNA splicing, sometimes different exons are spliced together, but if an exon's been not included that should be, then we're going to have a mutation occurring. Now, when a splice site mutation happens, the messenger RNA that is created is left non-functional. We're going to have quite a significant chunk of code that is going to cause our polypeptide chain to be wrong, and therefore our protein to fold incorrectly, and therefore we've got a non-functional protein. A good example of a disorder that's caused by a splice site mutation is something called thalassemia, and thalassemia causes hemoglobin, which remember is our protein that carries oxygen around the body, and it's found within the red blood cells, not to form correctly. So here you can see what is happening within thalassemia. So within thalassemia, we have within our abnormal splicing, that intron 1 is not removed fully. So with a normal splicing, the intron 1 is removed, and exon 1 and exon 2 are spliced together to produce our beta E globin protein. In thalassemia, part of that intron 1 is not removed, and therefore that interrupts that sequence, and because of this, that particular beta globin protein will not be produced, and it will break down within the cytoplasm, and that obviously is going to cause a problem in producing functional hemoglobin. And so this is the effect of thalassemia. So normally within someone who has the functioning genes, red blood cells are created correctly, they have the hemoglobin to carry the oxygen. In someone with thalassemia, that red blood cell is not going to be able to carry oxygen effectively. And I have linked to another little video clip in the Google Classroom, and you're quite welcome to click on this as well in the Google Slides. A link to a little video clip of a young gentleman who's giving details about how thalassemia has affected him and the effects of that particular genetic disorder. Now, lastly, I want to remind you that mutations are not bad news, okay, all of the time. Remember that mutations will be the source of new variation, and they're the means by which new alleles of genes arise. Remember, alleles are different versions of genes. Without mutations, all our organisms would be homozygous and no variation would exist in the population. And it's really important that we have this variation so that process of natural selection can occur. I know we've spoke about at the very beginning that mutations are very, very rare. On even more rare occasions, those mutations are advantageous, and that is important because that is the first step in the evolution of an organism. So remember, don't fixate on the fact that mutations are all bad. Yes, we're sometimes creating a non-functional protein, but that might give an organism a characteristic that helps it survive better in the current environmental conditions. So just to recap, what we have looked at this lesson is we've been looking at what a mutation is. We've taken a look at the 3 different types of gene mutation, and we've had a look at what effect those 3 different gene mutations have on the polypeptide chain and then ultimately the protein produced. I want you to remember the names of those 3 different types of gene mutation, so remember SID, and be able to describe what the impact of each of those mutations is on the protein produced. And then lastly, describe the effect of a splice site mutation, giving an example of a particular disorder that's caused by a splice site mutation. I have left some activities in the Google Classroom for you to have a little try at, so good luck with those.